Welcome to the Muscles Physiology PowerPoint. We're going to start this PowerPoint off by reviewing a little bit of the anatomy, just in case you've forgotten. And we'll start by talking about the three major muscle types, which include skeletal, cardiac, and smooth. Skeletal muscle is voluntary and attaches to bones. Cardiac muscle is found in your heart, and smooth muscle makes up your visceral organs. Now we're going to focus on skeletal muscle for this whole PowerPoint, but the mechanisms you learn in this PowerPoint are basically the same as the mechanisms that would power muscle contractions in cardiac muscle as well as smooth muscle. So the 6 functions of skeletal muscle is number 1, of course, producing movement of your skeleton. Those are your bones. When the muscle contracts, it pulls the bones in certain angles and directions. Maintaining your body posture and positioning. Although you don't put much thought into it, your muscles are contracted while you're standing up, allowing you to be erect. Number 3, supporting the soft tissues, like those in your abdominal area that aren't protected by a rib cage. You have several layers of skeletal muscle in that abdominal region protecting those internal organs. Guarding the body's openings, like the mouth, the vagina, and the anus. Those are all made up of voluntary skeletal muscles. Maintaining body temperature is done by working out the muscles, creating friction to release heat out of the body. Number 6, an interesting fact that not many people know is that skeletal muscle is a huge nutrient reserve. But what nutrients do we reserve in there? Well, we reserve calcium as well as a ton of glucose. So all of this skeletal muscle that we're going to see today are organized into a hierarchy. So the largest hierarchical classification is going to be the skeletal muscle as a whole. It is covered by a membrane called an epimysium. The whole skeletal muscle is made of smaller structures called muscle fascicles that are covered by perimysium. And the whole muscle fascicle is made up of smaller units called skeletal muscle fibers. Now what I want you to write down is that a muscle fiber is the same thing as saying a muscle cell. The words fiber and cell are interchangeable, and I want to make sure that you guys understand that because I do use both terms on the exam. All skeletal muscles are vascular, which means they have blood circulation, and they are all innervated, which means the nerves send signals, which causes the voluntary contraction. These are those 3 layers of membrane that I talked about. The epimysium is the outer connective tissue layer. The perimysium is the inner connective tissue layer that surrounds each muscle fascicle. Primary to this layer is that they are where you're going to find your blood vessels and your innervation. The innermost layer that surrounds each individual muscle fiber is called the endomysium. And this will have, again, vasculature in the form of capillaries as well as nerve endings. What you'll find under the endomysium that makes it different from the perimysium or epimysium is that underneath the endomysium, you're going to have these cell types called satellite cells. If you recall from anatomy, satellite cells are found in muscles as well as the nervous system, and satellite cells provide support, nutrients, salts to the cells that are surrounding them. So the satellite cells here in the muscle are going to provide nutrients and help to repair any damaged skeletal muscle. So again, these are the 3 layers of connective tissue that surround each of these 3 hierarchical layers of skeletal muscle. Now, when these 3 layers get to the end of the muscle, so you're going to have the endomysium here, the perimysium here, and the epimysium here. They all gather and collect at the end of the muscle into this white structure, which you call a tendon. Now, do you remember what a tendon connects the muscle to? Tendons are connective tissues that connect skeletal muscle to bone. And that's what we have in this slide here. The muscle attachments. They are a combination of the endomysium, perimysium, and epimysium coming together into a bundle that we call a tendon attaching the muscle to the bone. Now, the bundle is going to be found predominantly around the appendicular skeleton, which again are arms and legs. But for places that have flat muscles and very thin, flat-shaped bones like the skull or the sternum, instead of having a bundle-like tendon, you're going to have a sheet-like tendon, which we no longer call a tendon. It's called an aponeurosis. It is a tendon, nonetheless, but it is very flat like a piece of paper and lays flat down on the bone to help connect the muscles, let's say, in your skull to the cranial bones that make up your skull as well. So with that being said, I'm going to just mention one little thing about skeletal muscle, and then we're going to go through the hierarchy of muscles once again. What I want to mention one more time is that skeletal muscle cells are also called skeletal muscle fibers. Those terms are interchangeable. Anytime you hear fiber, I want you to think cell. Now, some very characteristic facts about skeletal muscle fibers is that they are very long. They are made up of hundreds of smaller cells we call myoblast cells. Myoblast cells are the individual tiny embryonic cells that are going to eventually fuse and fuse and fuse together until they make a huge, long, mature muscle fiber. Now, because you had fusion of hundreds of these myoblast cells, that means all of their nuclei fused as well. This means that a mature muscle fiber that is very long is going to contain hundreds of nuclei, which we call multinucleated. Those cells in yellow that do not fuse with the myoblast cells are called satellite cells, and if you notice, they're found on the perimeter of the muscle fiber. Again, the satellite cells are there to provide nutrients and help with muscle repair after potential injury. Do you remember what these lines are called from anatomy? The dark and light banding pattern? They're called striations. We will touch on that today as well. So here we go in terms of the hierarchy of skeletal muscle. So we talked so far about the entire skeletal muscle, which is made up of muscle fascicles, and then the fascicles, which are made up of muscle fibers. We're going to go through that hierarchy once again. This is just an introductory picture. Don't worry about memorizing this. We will talk about this in just a few slides. So, how is skeletal muscle organized? The largest hierarchy is going to be the skeletal muscle as a whole. So the pattern is going to be the same. It'll tell you the name of the structure, it'll tell you the membrane it's surrounded by, and it'll tell you what it's made up of. So an entire skeletal muscle is surrounded by a membrane called an epimysium, and it's made up of individual subunits called muscle fascicles. So now we're going to take one of these individual subunits and look at that. So here we have a whole muscle fascicle. It's surrounded by a membrane called the perimysium, and I want you to notice that it has veins, arteries, and nerves. And the whole muscle fascicle is made up of these structures called muscle fibers, aka what? Muscle cells. That means each of these structures here, 1, 2, 3, 4, 5, 6, 7, 8. There are 8 muscle cells here. So we're going to take one of these muscle cells, aka muscle fibers, and look at that. Each muscle fiber is surrounded by an endomysium. Endo, that prefix in Latin, means inner. It is the innermost connective tissue layer on a muscle fiber. So this muscle fiber, this whole thing here, is one muscle cell. You can see there's a nuclei here, there's mitochondria there, but you also notice that there's a bunch of these little red dots. Those are a next level of subunit. A whole muscle fiber is covered with the endomysium, and it's made up of individual units called myofibrils. We're going to take one myofibril, this circle thing, which is this circle thing right here, and take a zoomed in look of it. This whole myofibril is covered by this blue net-like structure called the Sarcoplasmic Reticulum. You can abbreviate this as SR. It's like an endoplasmic reticulum, very similar functions, but it's called a sarcoplasmic reticulum because it's found in skeletal muscle. Now, this myofibril if you look on the inside of it, do you see the red lines and the blue lines? Those are two different types of proteins. The red lines are called actin. The blue lines are called myosin. We're going to talk about in just a moment. But what I want you to notice about actin and myosin is that they're separated by these zigzag lines. So from this zigzag line here to this zigzag line, and I'm going to get my pen out and draw it for you. So from here to here, this whole thing is called a sarcomere. So you'll have this sarcomere, this unit, repeating itself back to back to back throughout the entire length of the skeletal muscle. We call these zigzag lines Z-lines. So with that being said, we're going to now go through and talk about the individual cellular structures of each muscle fiber, aka a muscle cell. So let me go back one slide. We're going to look at the structure of this guy and this guy right now. So I'm going to point something out before we move forward. This endomysium is connective tissue. It's this structure here. You see how it looks like kind of like a net? There's lines around it like that, like that. That's the endomysium. This membrane here, that pink one, that's the cell membrane of an entire muscle cell. Remember, all cells that exist in this world all have a cell membrane. You can call it a cell membrane or a plasma membrane. They're made up of a lipid bilayer. But because it's found in a skeletal muscle, we're not going to call it a cell membrane. We're going to call it a sarcolemma. So the sarcolemma is the name of the cell membrane that's surrounding a muscle fiber, aka a muscle cell. Inside, normally you have cytoplasm, right? All that yellow stuff is like cytoplasm. But again, because we're talking about skeletal muscle, we're going to call it sarcoplasm. So you can probably assume the prefix sarco has something to do with muscles. So sarcolemma, cell membrane. Sarcoplasm is the cytoplasm in that skeletal muscle cell. And what makes these guys special is that the sarcolemma is unlike a regular cell membrane. But why? What special qualities does it have? This right here. This type of cell membrane is able to transmit something called a transmembrane potential. This is a fancy schmancy word for voltage. Now, if you recall anything from the nervous system in anatomy or in the PowerPoint that I posted, in order to get a skeletal muscle to contract and move, you need to send it a signal from your brain through a neuron, and that's going to be a voltage signal. So once that voltage signal, let's say I'm going to draw a muscle cell here. Here is your brain, okay? And we have a neuron that starts with a cell body and comes all the way down to your muscle. And there's the synaptic terminal right there. Neurons are able to send signals from the brain to other places in the body using voltage. So this line right here, it's like my electrical voltage, and it shoots down. This is called a potential. The word potential means voltage in physiology. Once this voltage touches one part of the sarcolemma, it shoots to the right and it shoots to the left, and it shoots that voltage all throughout the sarcolemma, which is great because even though you're just sending a signal in this region of the skeletal muscle cell, you're able to transmit that voltage all across the muscle cell to get the whole thing to contract at one time. And again, that brings us to our second point. The ability of the cell's sarcolemma to transmit a voltage or a potential, aka potential, allows that cell to all contract simultaneously to generate the force you need to, let's say, move a table. So that is your sarcolemma. Now, there's another structure called T-tubules. And before I go over this slide, I'm going to go through the picture of T-tubules in the sarcoplasm on this next slide. So again, this whole thing is the sarcolemma. It surrounds the entire skeletal muscle cell. Let's just draw it all around. And this sarcolemma, if you notice, there's a hole there and the hole there. The sarcolemma is continuous and it comes down into this little pipeline. This pipeline that goes into the yellow, these yellows are called T-tubules. They're just continuations of the sarcolemma going deep into the muscle cell. Because sometimes your skeletal muscles, here's a skeletal muscle cell here, sometimes they're really thick. And if a neuron comes and sends a signal to the outer surface, you don't want that voltage to just get to the little pieces of contracting units on the surface. You want this voltage to go deep, deep, deep into the core of that skeletal muscle, so all of these units contract together. Your goal here as a human being is to signal contraction of a muscle and make sure the entire muscle, the surface of it and the core of it all contract. So how do you transmit a voltage from the surface to the core? Well, the voltage travels across the sarcolemma, down the T-tubules, all the way until it gets to the core of that muscle. So essentially the job of T-tubules is to transmit the voltage, which we can term an action potential. This term just means the voltage that travels through a neuron. You can transmit that action potential to the sarcolemma, then to the T-tubules to do this function. Make sure the whole muscle fiber contracts simultaneously. So once again, you have the voltage from a neuron from your brain. Comes, sends a signal to the skeletal muscle cell. That signal travels across the sarcolemma, down the T-tubules, till it stimulates the core of that muscle cell. Now, not only does this signal travel down the T-tubules, but we're now going to incorporate how it interacts with the SR. The SR is this blue net-like structure, okay? On the right and the left of the T-tubules, you have this thick portion here, you have this thick portion here, and here. Those thick portions are called terminal cisternae. The whole blue structure as a whole is called the Sarcoplasmic Reticulum. So, I'm repeating this a bunch of times because you have to know it. The voltage travels down the sarcolemma, down the T-tubules to go deep into the core of the muscle cell. You're transmitting voltage or an activation signal to the core of the muscle cell. But it's also going to travel right and left to the two cisternae. That voltage travels through the cisternae and then through the rest of the net-like structure. So now you not only have voltage traveling across the sarcolemma, down the T-tubules, but you also have voltage traveling all through the cisternae and the SR. Now, why does that matter? Why does it matter that we have to transmit that voltage down the T-tubules as well as to the cisternae? Well, the cisternae has a huge job. Its job is to store and release calcium. So this compartment here and this compartment here, as well as this compartment here and this compartment here, I just circled your terminal cisternae. Their main job is to store calcium. Make sure you know that. Calcium is a huge ingredient in allowing for a muscle to contract. Here's the bottom line. If you don't have calcium, your muscle's not going to contract, no matter what you do. So, the voltage travels across the sarcolemma, down the T-tubule, across to the cisternae, and it tells the cisternae to release the calcium. And that calcium will then go down into the actual proteins of the muscle to allow them to contract. So I think the biggest point here is that voltage traveling down the sarcolemma will travel down the T-tubules to make sure all units that are deep in the core of the muscle to contract, but it also travels to the cisternae to signal voltage-gated calcium channels to open. So let's write that down. Voltage-gated calcium channels to open. So if you recall from the mechanism of, the beta cells from the last PowerPoint, they had voltage-gated calcium channels. And when voltage stimulated it, it opened calcium channels, and calcium moved from areas of high concentration down to areas of lower concentration. So essentially the calcium left the cisternae and sprinkled down into these blue and red proteins that are inside. So far you've learned about how the voltage, aka the signal, travels from the sarcolemma down the T-tubules across to the cisternae to release the calcium out into the rest of the SR to get sprinkled down into the actual proteins of the muscle. Now, the structural components of the sarcomere, so what we're going to look at now is the individual pieces that make up the sarcomere, like the proteins, like actin and myosin. We've got proteins that are called titin, structures of the sarcomere that you have to know. This illustration that we have right here, and let me get my laser pointer again. This is the picture of the sarcomere from one Z line to the other Z line. In these red thin lines, you have what's called actin. In the blue thick lines, it's called myosin. Actin and myosin are termed myofibrils. Myo means muscle. Fibrils means long proteins. And those are the two myofibrils, actin and myosin, that make up the entire sarcomere. The green one you see here, this is called titin. We'll talk about that in just a moment. Now, to kind of touch on something you learned in anatomy, the dark and light banding pattern of skeletal muscle, which is called the striations, as you can see here, you have light, dark, light, dark, light. It's because of the regions of overlap of actin and myosin. So here you just have actin. Here you have actin and myosin, so it's darker. Here you just have myosin, lighter. Actin and myosin overlapping, darker. So the places where you have overlap or where you have a single protein is going to just dictate which one is your dark and which one is your light band. So again, the proteins are listed here, actin and myosin. The actin protein, the little thin red one here, it's literally also termed the thin filament. The myosin is the thick filament. So thin and thick filaments are going to be the two myofilaments that actually make up your muscles as a whole. And remember, all proteins are made out of amino acids. So when you're trying to build more actin or build more myosin, you need more amino acids. This is why people take amino acid supplements when they're lifting weights and working out, because essentially what you're doing is you're tearing the muscle and rebuilding new muscle, and you'll need amino acids to rebuild these protein filaments. Now structurally, I'm going to use a very similar picture, and I'm not going to read over this right now. You can read over this on your own. I'm going to go over it verbally with this illustration with you. So here from one Z line to the other Z line is one whole sarcomere. So this whole thing is a sarcomere. The thin filament is called actin. The thick filament is called myosin. We do have regions of a sarcomere. For example, the length of an entire myosin from here across to here, so this whole length is called the A band. It is the length of a thick filament. The open space between the thin filaments of actin is called the H-zone. The zigzag lines are called Z-lines. And the distance from the tip of one thick filament to the neighboring tip of this next sarcomere's thick filament is called the I-band. Right now, I'd like you to circle the I-band and the H-zone. Circle them and write a little note for yourself. I want you to know that these two regions shorten during a muscle contraction. So this muscle that you see here, this sarcomere, is a muscle at rest. How do I know that? Because when the muscle contracts and this Z-line slides in towards the middle and this Z-line slides in towards the middle, it causes this H-zone to get substantially smaller, and it squishes the I-bands of neighboring sarcomeres together. When you have that type of contraction, the whole point is to slide in from the right and left sides towards the middle of the sarcomere. The region that's in the middle of the sarcomere is called the M-line. So again, we have A-band, H-zone, Z-line, I-band, and M-line. Make sure you know what those represent. And on the previous slide, they're actually typed out for you. I'm going to go ahead and skip this slide because we already went over muscle striations. It is the overlapping actin-myosin patterns. And then this slide just repeats the contraction physiology of you causing a contraction to happen voluntarily in a skeletal muscle, and that signals the sarcomere to kind of squish in towards the M-line and become shorter. So this Z-line connected to all of its actins slides into the middle. This Z-line with all of its actins slides into the middle. In this illustration is one of the first times you'll see the titin drawn in. And if you look at it really close up, you see it's kind of a coiled pattern. This coiling allows us to squeeze in both sides towards the M-line, and for that to actually recoil back out to the relaxed state. So with that being said, I'm going to pause here and we are going to continue this portion on the next video.